Hi, my name is Roman Bond, and this is my research proposal. Research question: can Huntington's disease be prevented by fetal gene therapy? What is Huntington's disease? Huntington's disease is a fetal genetic disorder that causes the progressive breakdown of nerve cells in the brain. This disease can affect the striatum, which is a part of the basal ganglia located in the anterior region of the cerebral hemisphere. The basal ganglia controls voluntary movement, thought, perception, memory, and emotion. What is fetal gene therapy? Fetal gene therapy uses the intracellular delivery of genetic material for the treatment of a disease. Why am I studying this? No cure has been successfully created. Cures for Huntington's disease have already been developed but not properly tested. I hypothesize if fetal gene therapy is done when signs are caught early, the disease can be prevented. This study will consist of 6 randomly selected males and females that show early signs of Huntington's disease. The sample will consist of approximately 2 children, the ages 3 and 5, 2 middle-aged adults, ages 42 and 50, and 2 seniors, ages 65 and 70. The information to participate in the study will be posted in hospitals and online all around the U.S. and will be recommended by doctors to potential subjects. Procedure: this study shall take place in a lab setting, and each subject will be given a consent form to sign that gives the researcher legal authority to perform each procedure. When performing striatal neuroblast therapy, the cells are made from transplanted tissue, consist of dissected pieces derived from the lateral portion of the lateral ventricle of a fetus, embryonic cells, which are donated to the fertilization clinic for research purposes with informed consent of the donor. These cells would be injected into the human striatum, which is a part of the basal ganglia that controls motor and reward system. During cell treatment, stem cells would be transplanted to the diseased part of the brain. After reaching the affected area, the stem cells will produce new neurons and glia. In order to do this procedure, a magnetic resonance imaging machine will be used to pinpoint the damaged part of the brain. Then a surgical marker will be used to draw incision points onto the skin. When this is done, the subject will be placed under anesthesia, and a scalpel will be used to make an incision to the marked areas to expose the skull. Once the skull is exposed, a cranial drill will be used to make a burr hole the size of a dime over the striatum. After this is done, a syringe will be used to inject embryonic stem cells to the striatum. After everything is complete, the burr hole and incisions made on the skin will be sewn back together with sutures. Materials needed for this will be a cranial drill, scalpel, 32mm syringe, sterile regular tip surgical skin marker, functioning magnetic resonance imaging machine, absorbable sutures, cranial sutures, and scissors. Outcome measures: participants will be kept in a hospital for 1 year after the procedure and watched very closely. During this time, transplanted cells should form new neurons and glia cells. Participants will have daily blood tests and will be checked once a week to see if any new neurons form by being placed under fMRI. When participants have shown progression and healthy neurons are being generated, they will undergo rehabilitation. The participants will be taken through physiotherapy to help improve their movement. This therapy may consist of stretching to keep muscles from getting tight, straightening muscles that helps with posture, balance exercises, and wheeled walkers to prevent falls. When physiotherapy is successfully completed, participants are released and allowed to go home. Analysis: to collect data, each participant will be given a consent form to sign that gives the researcher legal authority to perform each surgical procedure. The participants will be told the benefits and risks of the surgical study. The participants chosen will be those who are showing early developments developing symptoms of Huntington's disease. To analyze the data acquired, the researchers will use a chi-squared test. This test is any statistical hypothesis test where the sampling distribution of the test statistic is a chi-squared distribution when the null hypothesis is true. Discussion. If this study shows that fetal gene therapy can cure Huntington's disease, then this would be a groundbreaking breakthrough for future research. This research could lead to the disease being cured before symptoms occur. If the research shows no progress, birth defects in future generations can be a major problem because the scientific aspects of human genetics are still poorly understood.